Gathering with large groups of people in small spaces for prolonged periods of time is basically what I used to do. It's strange to think that something that seemed so harmless is now potentially lethal. I keep thinking about how many microbes we used to exchange when we gathered in theatres and cinemas. A whole other drama going on unnoticed around us. And I wonder how the microbiomes of our streets and ourselves will change. Whole cities sprayed with bleach, and our hands scrubbed and stripped of all their inhabitants. Making performances is a collaborative act, but working with other people is impossible in this particular moment. So I want to try and shift the scale. In my isolation, can I see myself and the space that I'm confined to as a collective? Can I imagine collaborations with the different organisms that are part of me and my environment, staging disagreements with my mitochondria, my organelles, my E. coli? If this was a play, could I have all the characters I needed at my fingertips? I can't do what I did because of a virus, so I wanted to see if a virus could allow me another way in. I want to begin with a virus called T7. Bacteriophage T7 is a virus that infects the E. coli bacteria. And this is the structure of its DNA. The ATCGs that you see here are called nucleotide bases, and they come in pairs, A's with T's and C's with G's. When you see a red bar underneath the line of base pairs, that indicates a gene, which means that these letters code for a particular protein. T7 is just under 40,000 base pairs long, and it has 52 genes. It's so simple an organism that scientists describe it as barely alive. According to the central dogma of biology, DNA is transposed to RNA, and RNA is translated into proteins. You can think of this process like a performance. It's basically Brecht's theory of epic theatre. DNA is the script or score, notations on a page. RNA is the transposition of this to the words or gestures of the performer. And the final stage, the proteins, are the transformations these gestures affect in the audience. This particular organism, T7, is also the beginning of a story. The story of synthetic biology. It begins with an engineer named Andrew Endy. For his PhD, Endy decides to make a computer model that will predict the changes of behavior in an organism when the order of the genes is switched. He chooses our bacteriophage T7 to model. He spends the next 7 years trying and failing to make a model that will accurately predict the workings of the simplest and most studied organism that exists. When he fails, and this is the crucial part, rather than revise the model, he decides to revise the organism. This counterintuitive leap, this jump from descriptive to prescriptive, is the foundation of the greatest revolution of our time. Just think for a minute about the breathtaking reversal of logic required to make that leap. You don't fit my predictions, so rather than revise my predictions based on your behaviours, I will revise you to fit my expectations of how you should behave. To revise the organism, Endy cut out the so-called junk DNA, that's DNA that doesn't code directly for proteins, and separated out overlaps, added cut sites. He simplified and he removed complexity. The revised organism was hyper-effective in the new environment it had been engineered for, the lab. But the kicker? When compared with T7 in doing what the bacteriophage normally does in the wild, which is to infect E. coli, it was hopelessly inefficient. By removing the complexity, Endy had removed what made the bacteriophage function. There's a similar story with human DNA. When the Human Genome Project first sequenced our DNA, they were surprised to find that more than 85% of it was not protein-coding genes. Some of it was even DNA from viruses like T7, which had been incorporated into our genome long ago. These scientists had been hyping it up as the script for life that would give us the key, tell us who we are. And then they found out that more than 85% of who we are was, to them, nonsense. Adorno, the untruth of identity, the fact that the concept does not exhaust the thing conceived. Something hopeful here, in the idea that the thing conceived will always be more than the word for it can hold, that the world will never be completely overwritten by the human mind. Otherwise, could you imagine? Words would burn ideas right up. If we think of the DNA as a script or a score, what kind of performance does it code for? Let's try a very simple experiment, an analogy for the simplest gene. I write the line, and you see it on your screen. That's the messenger RNA. Then you, the audience, turn it into actions. Those are the proteins. Let's try it. And then I write, that's a slightly more complex one, where things are open to interpretation. And then I write, or how about if I write, do you notice how many variables I had to take account of in that set of instructions? Your genome has to make these allowances too for the different cells and what they're there to do, so that the cell in the arm will disregard the instruction to make synapses, but listen to the one for making skin. And then I write, or how about if I write, but now I'm being like Endy, thinking DNA could be so simple. Like Brecht never told anyone what action to take. He set up scenarios, as in a petri dish, and then asked the audience to observe as the experiment played out. He doesn't provide instructions for what actions to take in the world. He just shows you that the world needs changing.